Since ancient times, every living creature on planet Earth has gone through evolutions of structural and cellular development to build immunity, in order to survive the ever-changing environment, and to be able to prevent diseases caused by the pathogens. Like invertebrates, or animals that lack a vertebral column or backbone, such as insects, have outer shells that we call exoskeleton, made from chitin, which is a polysaccharide present in the exoskeletons of arthropods. As a result, the shells are strong and can act as an effective outer shield. Other than this, there are enzymes called lysosomes in the intestines that destroy food that is contaminated with bacteria or pathogens. And this species also possesses hemocyte cells that use a process called phagocytosis, an action of engulfing pathogens with its plasma membrane to destroy invaders such as Plasmodium, a protozoa that causes malaria in humans. Now that we know about the immune systems of the invertebrates, what about the vertebrates?
Vertebrates are animals that have a backbone inside their body. For example, humans. This is Sarah. Sarah is a human. In her human body, there is a defense mechanism to defend pathogens from entering the body. This mechanism is called the first line of defense, present in different body systems, such as sweat glands, oil glands, stomach lining, urinary bladder lining, and birth canal mucosa, have specific substance that can capture and inhibit the growth of pathogens. In trachea, a part of the lower respiratory tract, there are cilia and mucus to trap foreign particles like antigens. And as a result, those antigens will be swept out of airways and out of the body in the forms coughing and sneezing. However, what if the first line of defense wasn't able to defend all the antigens? Don't worry, that's why there is the second line of defense.
When a wound occurs, the body may be approached by the outside invaders. Luckily, we've got the Avengers. The second line of defense. Here comes the members. First ones, the brother phagocytes, neutrophil, and macrophage. Their job is to distinguish the invaders by phagocytosis. Second one, dendritic cell. Securing the surface tissue, it also engulfs the invaders with their plasma membrane and activates the specific defense mechanism. Following up, the foreigner's detector, eosinophil. Guarding under the epithelial cell, once it has detected the target, the parasites, it'll have a specific enzyme secreted to save the day. On to the next member. We have basophil, who secretes histamine to stimulate the process of the immune system. Finally, natural killer cell, who actually is a natural killer. It secretes chemicals which stimulate apoptosis, the process of programmed cell death, to keep our body remain unconquered.
Here are the body response when approached by outside invaders. Mast cells will signal macrophage with histamine for help. Once it arrives, no foreigner will be left alive. Also, basophil and mast cells have their histamine secrete to cause vasodilation, the widening of blood vessels, which is white blood cells and lymph way to the wound. This process makes the wound swell and body temperatures arise. Aha! White blood cells are here. Bind the uninvited guests and farewell the damaged tissue. The pathogen. Tissues and the warriors who sacrifice white blood cells will then turn into abscess and get eliminated through the wound. The mechanisms mentioned previously. Inflammation is non-specific defense mechanism. Then, how about specific defense mechanism? The invaders not only activate non-specific defensive mechanism, but also stimulated specific defense mechanism. Let's see how.
Third-line defense is different from the first and the second as it applies to adaptive immunity, aiming for specific targets. And the white blood cells that will play an important part here are lymphocytes and antigen-presenting cells. In this section, the term antigen is used for calling pathogens that trigger adaptive immunity. Something like criminals with a bounty on their heads. Macrophage cells will get rid of pathogens by phagocytosis. After digestion, fragments of the antigen are left. These fragments are brought to the protein on the cell surface of a macrophage called Major Histocompatibility Complex Class 2, or MHC Class 2. The main purpose of this process is to induce lymphocytes and other cells' activities. We call this macrophage antigen-presenting cell, or shortened as APC. To make it clear, lymphocytes are a group of white blood cells with a big nucleus that takes up pretty much the entire cell space. They can be categorized into two groups, T cell and B cell. The first, T cell, is a lymphocyte that grows from bone marrow, but later comes to full growth at thymus gland. The second one, B cell, grows and then matures in the bone marrow.
T cells have an antigen receptor called T cell receptor or TCR on their surface that is specific to the antigen. The TCR's limb is positioned to attach to the APC-presented antigen fragment. There are two types of T cells that are very important in this process: helper T cell and cytotoxic T cell. Helper T cells will capture antigen fragments presented by APC and produce a signaling chemical known as cytokine and cause other T cells and B cells to respond to pathogens. When stimulated by helper T cell, cytotoxic T cell will increase its number by cell division. And now, cytotoxic T cell is ready to fight. After encountering the infected cells, it will release an enzyme, perforin, to penetrate them, and also release granzyme into the perforin pore to stimulate apoptosis, causing the infected to pop. B cells have an antigen receptor called B cell receptor or BCR on their surface that is specific to the antigen. The BCR's limb has two areas that can attach to the antigen. When the antigen attaches with BCR and acquires cytokine from the helper T cell, the B cell is stimulated to proliferate. Some will grow in size and change into plasma cells, which play a role in generating antibodies. And these antibodies will collect antigens, for it would be easier to be disposed of by phagocytes. Other B cells will develop into memory cells, which are very helpful as they can detect and get rid of the same antigens if they were to invade our bodies again.